Hello, everyone. Oh, sorry. My name is Sharon Istvanfi. I'm a PhD student in Marc Somoza's lab. And I'm going to show you now part of my PhD thesis, hopefully, which includes optimizing composite DNA letters for high-density information encoding. Like this, it works perfectly. So first, let me tell you more about our photolithographic synthesis on the machine that we're using, the maskless array synthesizer 2.0. I know Hamid already gave you a great introduction to it, but let me tell you again, we're also excited about this machine, so why not repeat it? So as already said, it's a photolithographic synthesis, which means that our most important step, or the important step that is different from chemical synthesis or electrochemical synthesis, is the deprotection with light, so with the UV irradiation. How can we do that? We are not using DMT-protected phosphoramidites, but we're using NPPOC derivatives, which in this case we're using now benzyl NPPOC, which has a higher quantum efficiency than the normal NPPOC, which just means that we need less light to effectively cleave it off from our phosphoramidite, the building block for the synthesis. So this is all the chemistry you will see after it. It won't be as much. I just wanted to show you that through light we get an excited state of the photolabile group that then leaves afterwards, and then we have here, in the end, through multiple following reactions, our deprotected nucleoside phosphate, which is now free for another coupling reaction. Now let me show you our machine, and this is the optical system. We have our UV light source that through which the UV light is then reflected onto the flow cell, and somewhere here we have our DMD, which is a digital micromirror device. It has a lot of different micromirrors. Actually, it has around, so for one of the DMDs, it has around 780,000 that we're currently using now in the lab, and for another machine has a DMD with around 2 million micromirrors. And what's great about these micromirrors is that you can individually change their angle. So individually we can change through digital masks if light is reflected onto our glass surface here or not. So for these three mirrors, the angle is on. So there, the light is reflected onto the surface. So only where the area is illuminated by the UV light, deprotection happens. This is very important to understand, so you can understand our synthesis in general, because only through that we're able to do the light-directed massively parallel synthesis. So let me walk you through this. We have a photolabile protecting group sitting on each of the phosphoramidites. Now we're shining light onto, for example, position 2 and 4. The photolabile group is removed by that. We add our next building block, G in this case. It couples at these positions, and then we repeat it many, many times, and we get different oligos. So it looks a bit like Lego. This is how the machine looks in real life in our lab. And now let me walk you through the laboratory process to synthesize a library. So first, of course, there is the synthesis and everything, all the steps after it, like base deprotection and all these things. Next, we are cleaving our library off of the surface. We're doing that with water because we have a cleavable DT that has an ester group, and through water we can just cleave it off the surface. That's quite good because water is always good to use in the lab instead of something else. And next we have our library in the water. A lot of times we're also doing some verification that actually synthesis happened for non-cleavable sequences. We hybridize them with a psi-free labeled complement, and then we check, okay, the synthesis happened, that's good. And also from our library in the water, we're also doing some verification if it worked. For this, we're doing PCR and the gel electrophoresis. And thank you to Jenna, she already explained that, so I don't have to. And in the end, after library preparation and everything for sequencing, we get our sequencing results. So, let me tell you something about composite letters, because now you know how the synthesis works for the 4 bases and also how it works in general, but I want you to understand the idea of composite letters. First, why should we use composite letters? It's very easy. It just increases logical density, so it increases the information that can be stored for each cycle. At the moment, if we are using only natural bases, not using composite letters, we are able to store 2 bits per cycle, since we have an alphabet of 4 letters. But if we increase this alphabet from 4 to, for example, 8 letters, we are able to store already 3 bits of information for each cycle, for each building block. And that is great because just to give you a number, if we have a 60-mer, we're now able to store, instead of 120 bits, 180 bits, which is a 50% increase. That's already great. And if we're able to do even more, like in Hamid's talk, then we'll be even better. So this idea was proposed in the paper by Leon Anavi and Zohar Yachini. So I would invite you to read also this paper if you're interested more in the theoretical part of it. And let us take a look at the composite letters now. So this is one building block that consists of a mix of, for example, here n of 4 of the 4 bases, equimolary. And then we also can, for example, have a building block of only 2 bases. But also we can have non-equimolar mixes that are, for example, 70% E and 30% A. And in the lab, how we do it, we just mix it by hand. And so there it's pretty easy. We can just mix 2 or more phosphoramidites, and also we can mix the ratios as we want. With weighing and everything, we can calculate the ratios. And then we are able to add our mixes for now. So this is the machine we're using now. There are also other expedites that have more ports. But for ours, we have 4 empty slots. And so we can add 4 composite mixes. But the machine also has automatic mixing, the ability to automatically mix. It has the IUB letters implemented in the program. IUB letters are equimolar mixes of all possible combinations, 2 component mixes, 3 component mixes, and then n. Now, let me show you some of the results. First, I want you to see the sequence design that we have in general. So here is the glass surface, then we're using a DT linker. We have a cleavable DT, and from this point onwards, everything will be cleaved into the solution, into the water. We have our adapters, which are important for the sequencing. We have our barcode, which is important to distinguish each variant from each other. And we have our, I called it information containing region. So this is the region where information could be stored. So to show you one of the variants, one of the analysis of the variants, so we sequenced this with an Illumina sequencer. And these were the filtering conditions for the analysis. We filtered for a barcode edit distance of 0, so 100% overlap of the barcode. We also filtered for the full length of the sequence and for an alignment score of 30. And here you can see, so these were all automatic mixes. And here you can see the ratio it should be, and here you can see the actual ratio that we got from the sequencing results. So we used 2 component mixes, R, Y, and M, and also we used N in this case. Now, if we take a look at all of the composite letters that were mixed through the machine, we can see most of them deviate much from the predicted ratio, which should be equimolar. And we have quite high deviations, which is expected due to the machine just. So, and the deviations also are higher for 3 component mixes, also for N in the range of 30%, then they're higher than for the 2 component mixes. Also the maximum deviation, so this is the maximum deviation from the predicted result, since we had more than one position where we had a composite letter, we had some that had this high of a deviation from what we wanted to see. But, interestingly enough, we're still able to get, through predictions, most of our sequences correctly. And we have around an error rate of, let's say, 1 falsely predicted composite per sequence. And yeah, actually also with higher alignment scores we had better predictions, but I wanted to show you this because here you can also see some of the, more of the substitutions and everything. So next, let's take a look at manual mixes, in this case for 2 different ratios. So these are 1/3 and 2/3 CET mixes, and the other way around. And here you can also see, again, an example sequence. And the filtering conditions are the following. Again, barcode edit distance 0, full length, and alignment score of 20. And here you can see it looks already more as expected, or more as we wanted it to be. And also in general, we can see that so this is called highest deviation because I wanted to see, since we also have gaps, it's the highest deviation from the both components for the average. So in this case, T has a higher deviation from the average than C, but you can still see it's a lot lower than for the automatic mixes. Also the maximum deviation, which we had for E at the position 41, which is 70.44%, is also much lower than for the automatic mixes. So here I have the equimolar mixture of C and T, which is called Y, and it had 12%, which was still quite good actually for the automatic mixes. And so, I mean, as expected. But still we were able to get our predicted sequences. And finally, I want to show you a sequence where the differences between, so we have 4 different mixing ratios for C and T, and the differences of each of them are just between each other 10%. And also here we were able to get, so this is the designed sequence, and we were able to predict the correct sequence in the observed. So with this, I would like to thank the whole team of the DIDEX consortium, especially all the informaticians who were working with me on the analysis. And also I would like to thank my team at the LSB, my colleague Hamid, and of course Mark Somoza, who's my supervisor. And I would like to thank you, the audience, very much for your attention.
So thank you very much, Sharon. Do we have some questions, comments, notes for Sharon? So if there are no questions, thank you very much, Sharon.
Thank you. Oh.
Ah, okay.
Thank you.
it's related. It's related question I asked before. Do you have an idea of the error rate you get when you make these mixes, when you sequence? Is it difficult to really identify? You show some deviation. So what would be the expected error rate you would get with these mixes of different letters?
Yeah, so that's a question that we also have and that we also try to answer. It's a bit difficult since of course, already aligning the sequences introduces error rates that we introduce gaps at positions where maybe there aren't gaps because, the alignment algorithm prefers gaps at certain positions, maybe more in the beginning of the sequence than at the end, especially if we look at sequences. We want to show this, especially if we look here. So this was a sequence that had, in this case, C, T, and then a mix of C and T. So who knows if the deletion comes from, for example, F or from this position at T. So yeah, that's a question that we're also looking to maybe answer next time, I hope.
Okay. I also have another question on your DMD device. Do you have really perfect separation on each synthesis site, or do you have some cross-contamination with neighboring cells?
Oh, so wait, I would just show you the DMD here.
Yeah.
So we have here our mirrors that are, each of them have the size of 10 by 10 micrometer, and in between we have, not sure, but we have around 1 micrometer of space in between them. And yes, there are some insertion errors from the light reflected onto the, but it shouldn't be too much since it's 1 micrometer in between them. But also we're trying to quantify these errors with sequencing results. So there's a paper already quantifying some of these errors. It's called, it's by Jody Littard and Mark Somosas. So they already did some quantifications. We also want to quantify it even more with sequencing results later. But yeah, there are errors from, yes.
Thank you.
Thank you.
So, another question there.
Thank you for your presentation. The question I had is, do you know the efficiency of activations when you apply the UV? Because it seems that you have several strands that you will activate per spot. So do you have activation at 100%? And the other second question is, do you do it in aqueous solutions or is it dry?
Oh, okay. So I didn't catch the first part quite well, but I think if I'm correct, you asked me about the efficiency of the photo cleavage.
Yes.
Right, okay. So it is higher than if you have more light, it gets higher. So that's just in general something, of course. And how we use it now, as far as I know, it's 99% for ACT and around 95% for G, if I'm correct. Yeah. So this, and the second question
is it so because you need to remove the byproducts? So is it in aqueous solutions or? Do you
yeah, you asked. No, we're so here the photo deprotection is in DMSO, and our reaction cell. Oh wait, I want to show you. Our reaction cell is always floated with some reagents. We have a drying step in between, but other than that, we always have either ACN or some reaction, solvent for, reagent solvent for, yeah.
Thank you.
So, thank you again, Sharon.
Thank you very much.